So the last section in chapter 7 is about bulk transport. And this is when we involve actually the plasma membrane, not just proteins in the plasma membrane, but we're actually actively using the plasma membrane to move things across it. And this is very active transport, like I said, actively, because we need to put in not a ton, but we need to put in ATP to make this happen, because we're actually going to have the cell membrane reaching out and pulling things in. So we're going to need to put in energy for that to happen. And there's two ways that this happens, by endocytosis and exocytosis. And you guys can probably guess what those have to do with, because exo means out and endo means in. So exo is when we get rid of things through the cell membrane, and endo is when we use the cell membrane to take things in. So like it says, small molecules and water enter or leave the cell through the lipid bilayer or by using transport proteins. And we just went over that in section 7.3 and 7.4. But big molecules, such as polysaccharides, huge sugars. Think about it, glucose can't get across the cell membrane. How is a polysaccharide made of hundreds or thousands of glucose molecules supposed to get across the cell membrane? And also proteins, hundreds, thousands of amino acids. These have to find a way to get across the cell membrane as well. And this happens in bulk, meaning a lot of them, through the use of vesicles. So remember, vesicles are little portions of the cell membrane that float around the cell, and they carry things in them. So that's basically what we're going to use. This is vesicles either forming from the cell membrane or fusing with the cell membrane to get rid of things inside the cell. And this requires a lot of energy, so it's active transport. So exocytosis is the first one. Exocytosis is when transport vesicles, so remember they're moving all around the cell, carrying things wherever they need to be, from the ER to the Golgi, from the Golgi to the lysosome, or from the Golgi to the cell membrane, wherever it may be. They go to the cell membrane and they open up and they release their contents outside. And a lot of secretory cells, so like our insulin cells, our pancreas cells that make insulin, these use exocytosis to get rid of their products, because insulin can't cross the cell membrane, so it needs to get rid of it somehow. So the insulin is made somewhere, packaged up by the Golgi, and the Golgi ships it off to the cell membrane, and the cell membrane fuses with the vesicle and opens up, letting the insulin go outside of the cell. Endocytosis is exactly the opposite. Do I have pictures here? Okay. Endocytosis is when the cell membrane forms a vesicle by taking something in from the outside. Taking in macromolecules, very large molecules. And endocytosis is the reversal of exocytosis. So it's basically exactly the opposite, and it has different proteins. So remember, proteins are how we're communicating with everything. There's 3 types of endocytosis that you need to know about. Phagocytosis, which is cell eating. We've already talked about this one, when we form a food vacuole. And then there's also Pinocytosis, which is when we take in smaller molecules, liquids, so it's cell drinking. Or then there's receptor mediated endocytosis. This is when we have to use receptor proteins as a messenger to know what we're supposed to take in. So in phagocytosis, a cell engulfs a particle in a vacuole. So this is big, large food molecules. That's what it's taking in. And then, since this is such a large molecule, our cell can't use such a large molecule right away. So what it does is it fuses with the lysosome. The lysosome breaks down the food and then can send the subunits, the monomers, to wherever they're supposed to go to do their job. In pinocytosis, molecules are taken up when extracellular fluid is gulped into tiny vesicles. So basically, the cell membrane goes like this, takes in some extracellular liquid, that's why it's cell drinking, and inside that extracellular liquid are particles, molecules that the cell needs, that they can then use, and then they'll be shipped around the cell wherever they need to go. Lastly is receptor-mediated endocytosis. So this is taking in of something through the use of the cell membrane, endocytosis. But what happens is we get messengers somehow that it's going to happen. So there are receptors on the cell membrane, and they're only going to form a vesicle if something binds to that receptor. And this is what's called a ligand. So a ligand is any molecule that binds specifically to the receptor site of another molecule. So it's kind of like how a substrate binds to an enzyme. A ligand is something that binds to a receptor. So let's look at a picture. So here we're trying to take in these little green guys, and we have these receptors on the outside of the cell. So they're only going to form a vesicle if the ligand binds to the receptor, and then the receptor is going to cause the vesicle to form. The receptor tells these coat proteins right here to start forming a vesicle. So it's just a bunch of messengers. The ligand comes in, binds to the receptor tells the coat proteins to form a vesicle. Now we've taken in the ligand and the receptors from the outside and these green guys that we wanted. So you can see, this was a question on your last quiz. Here we have the proteins are on the outside of the plasma membrane, but they're on the inside of a vesicle from the way that they fuse. So receptor-mediated endocytosis is when we use a receptor to help us mediate endocytosis. This is regular pinocytosis, so the plasma membrane takes a big gulp of extracellular liquid, it drinks the extracellular liquid, takes it in along with everything that's out there. So we got purple guys and green guys, and now it's inside a vesicle. Cell-eating phagocytosis is when we take in a big piece of food or something. Then it's inside a vesicle, inside of our cell. We're going to send it straight to a lysosome to be digested, broken down for parts so we can use it for something else. This is an amoeba eating a bacteria. So this stuff around the outside is the amoeba, which is a kind of single cellular organism, and it's eating this bacteria, which is that long skinny thing in the middle. This is an example. This is a photo of pinocytosis. So this is the cell. This is probably a large vacuole is what it looks like. And then all those little inserts is the cell forming vesicles from the plasma membrane. And again, you can see here, this is actually a receptor-mediated endocytosis happening. So the coat proteins form along the inside of the plasma membrane and help the vesicle form. Then the plasma membrane is folded in, and we take all the receptors and the ligands in with it. And that is it for bulk transport.